In this video, we're going to be looking at the nucleus. So we'll put that as our heading for today, and I'm sure you've heard of the nucleus before, and this can be referred to as the brain of the cell. And just as the brain of the body controls the functions and the thoughts and our personality, the nucleus controls everything from the structure to the function of the cell. And there's normally one nucleus within the cell, but this can vary depending on the type of cell. So if we take a look at red blood cells, so RBCs, they actually have no nuclei. And this is in opposition to other cells, such as muscle cells and liver cells, for example, which have multiple nuclei. So multiple nuclei. So the nucleus, if we draw a cell, the nucleus is normally visible as this purple color under light microscopies using different stains. So it's normally this kind of round or oval shape. And the purple is the chromatin, which is visible. And we'll write that down just so you can see what that word is, chromatin. And we'll explain exactly what this is, but generally chromatin refers to loosely packed genetic material within the nucleus. And the nucleus, it houses the genetic material of the cell. So this is things like, and if you write these terms down, so we'll write some key terms. So this is things like genes, and we mentioned chromatin, and similarly we'll say chromosomes, which are just essentially more tightly packed than chromatin. Then we've got nucleic acids, which you may or may not have heard of, but you will definitely have heard of DNA and RNA. So that's just an example of some of the things that the nucleus contains. And let's take a deeper look at the structure of the nucleus. So let's write our heading down. Nuclear structure. And to depict this, we're going to draw two diagrams. So the first is just going to be a bird's eye view of the nucleus. And the second is going to be a cross section. So if we just cut into the nucleus like this, you can see some other aspects that we won't be able to see on the bird's eye view. So looking at the first diagram, we normally start from the outside of the structure. So the nucleus, actually, what it has is it has two membranes, very similar to the plasma membrane. If we remember correctly, plasma membrane has a phospholipid bilayer, just like the nuclear membrane. So the nuclear membrane is actually not what we're going to start from. We're going to start from further out. And we've got this structure here, which if we draw this in, it looks like some sort of a cavern or a matrix. And this is our rough endoplasmic reticulum, RER for short. And we've got our cisterna, which is the inner cavity of the rough endoplasmic reticulum, but we also have on the outside, we have these small components called ribosomes, which we'll remember are involved in the synthesis of proteins. And they actually continue all the way until the outer nuclear membrane. So it's continuous with the nuclear membrane. So we'll write in the names. So we've got our outer nuclear membrane. And when we've got an outer membrane, we've also got the inner nuclear membrane. And within both of these, or between both of these, you'll notice that there's actually a space. And this space is just simply your perinuclear space. So the outer nuclear membrane, the inner nuclear membrane, and the perinuclear space, they all form what is called the nuclear envelope. Okay, so the nuclear envelope is the outside of the nucleus, and it's continuous with the rough endoplasmic reticulum. But the nuclear envelope is not actually a continuous structure. Well, why is it not continuous, you might ask? It actually has, if you look at our second diagram, these small holes, if you like. And these small holes, they form around 10% of the total surface area of the nuclear envelope. And we'll draw these on our first diagram as well, just to depict this. They go all the way around the nuclear envelope. And we'll see that from our second diagram that it's essentially a hole which is surrounded by an outer ring. And we'll zoom in a little bit on this. So these structures, which are actually, if you label them, they are called our nuclear pores. Nuclear pores. Or they're also known as the nuclear pore complexes, NPCs. So we'll put NPCs in brackets. So let's zoom in on one of these. So we've got their inner hole, if you like, and this outer ring. Now this outer ring is composed of about 30 different proteins. And remember, this is in between the inner and the outer nuclear membrane. And these proteins, and attached to these proteins, are some other components. So we've got these kind of tentacles that are coming out, and this will be our this side will be the side of the cytoplasm, and this will be the nucleus. So the side of the nucleus, these structures, they're actually forming this kind of basket shape. And the name of these structures is actually simply just protein fibrils. So we'll call these protein fibrils. And their purpose is, so any molecule which is inside the nucleus and needs to exit, these protein fibrils essentially recognize it through a complex series of mechanisms and take it into the nuclear pore and then out into the cytoplasm. So these will be large molecules which are going through an active process and either leaving or entering the nucleus, whereas small molecules will just passively diffuse in and out. Okay, so that's the nuclear envelope and the nuclear pores. Now moving inside of the nucleus, what have we got? Well, we have a very interesting structure which is composed of intermediate protein filaments. And it's forming this mesh-like network inside. And you'll notice that one side is attached to the inner nuclear membrane. Well, what's the other side attached to? It's attached to this component, which forms the majority or a large amount of our nucleus. And we mentioned this before, this is our chromatin. So the other side is attached to chromatin. And what is this mesh-like structure called? Well, this mesh-like structure is, the name of this is just the nuclear lamina. Nuclear lamina. And these proteins that we talked about are called lamins. Simple as that. So the purpose of the nuclear lamina is just to give the nuclear envelope, the nuclear membranes, some sort of structure and to hold it together and stop it from collapsing in. Okay. Now the chromatin, let's talk about more about this in detail. So the chromatin, let's write about this here. So chromatin. Now, there's two types of chromatin that we want to mention. And one of these types, so number one we can say, is heterochromatin. Heterochromatin. And our second type is euchromatin. Euchromatin. And what's the difference between both of these types of chromatin? Why is it even relevant to this nucleus, the structure of the nucleus? Well, this chromatin, which is attached to the nuclear lamina, so remember the nuclear lamina here, this chromatin is actually heterochromatin. And the heterochromatin is more densely packed, is more densely coiled, and therefore has a darker appearance. Whereas the euchromatin is less densely packed, it's less densely coiled, and it has a lighter appearance. So for that reason, what you're going to find on the inside of the inner nuclear membrane is this dark appearance. And that's just because of the heterochromatin, which is attached to the nuclear lamina filaments, the lamins. And you will find some dense pockets dotted around as well. But largely what you'll have is you'll have euchromatin, which is this lighter colour, and this will form the majority of the chromatin within the nucleus. Now, saying that, the nucleus, like the cell, also has a fluid component to it, and this is simply called the nucleoplasm. So we'll label that and put that here as the nucleoplasm. And the last aspect of the nucleus that we want to talk about is, if you go to our second diagram, you'll see this densely coiled structure, and it's actually quite significant in size, generally housed around the center. This is called your nucleolus, and if you've got many, your nucleoli. And this houses, so nucleoli or nucleolus, and this houses various different things. So all the genetic material, so this will be ranging from DNA to RNA and to various different proteins. So there you go, that's a quick overview on the structure and the function of the nucleus.